to discuss on the topic sustainable management of solid and liquid waste. So it's a kind of a review work. So I'm not going to detail of the experimental details, but I want to discuss with you. So if you have questions, you can always ask questions directly to me. So and I'll be happy to answer those questions. So first of all we want to be rich in terms of financial aspects, and to become rich, financially rich, we what we do for the economic growth, we used to exploit environment. By degrading environment, by exploiting environment, we want to achieve our goal, that is the financial richness. So you can understand the natural resources are being degraded, are being exploited to meet our demand. We want to be rich. Resulting, what's the result? Resulting in a devastating effect on the planet. It causes global warming, it causes climate change and pollution. So the entire planet is facing a tough challenge to maintain a balance between the economic growth, economic well-being, and the ecological sustainability. Therefore, we got to optimize. So to maintain the balance, we got to optimize the exploitation of our environment, and so that the the ecology cannot be damaged, or ecosystem can be going on in a sustainable manner. So it shouldn't yield the productivity of the our ecosystem. So we got to maintain the ecosystem, the balance. So therefore we need to optimize the way of using our environment, and then we need to meet our financial demand. So the question is, what is sustainable development? Sustainable development is a development that meets the that meets the present need, and without compromising the ability of future generations to meet their need. So you can use the environment, but you shouldn't perturb, or you shouldn't compromise the ability of generation to exploit the environment for their need. So for the sustainable development, in one hand, we can improve lifestyles and our well-beings, but we should preserve natural resources and ecosystems. So this should go side by side. You cannot perturb one of them. So to protect the planet, United Nations in 2015 adopted certain goals, sustainability development goals, that include like no poverty, no hunger, good health and well-being, etc., etc., to protect our planet Earth and to ensure that all can live in health and prosperity. So these are the three main pillars of sustainable development: environmental protection, social development, and economic development. In one sense, they are independent. However, they are mutually reinforcing. And out of these three, environmental dimension has been recognized as the most important one. And the dominant environmental issues include air water pollution, climate change, loss of biodiversity, and land degradation. So today I'll be focusing mostly on environmental pollution. So environmental pollution, as you can understand, is a global threat with respect to human health, with respect to environmental health. It can damage ecosystem, it can cause loss of biodiversity, and with regard to the human health, as you can imagine, whenever we are exposed to environmental pollution, it causes stress. Stress can uh, cause the immune system down, resulting in microbial infection, resulting in many inflammatory diseases. So that's how it is a threat to human health, it is a threat to environment. So since pollution perturbs ecological balance and threat to human health, eco-friendly approach, eco-friendly remediation of environmental pollutants is absolutely necessary. Now you can see various types of pollutants in these slides. Uh, one of the important pollutants, that is plastic. And so I am taking plastic as an example, but it shouldn't be restricted to plastic. You can imagine all sorts of pollutants, and you can also imagine their remediation in different ways. Taking plastic as a model system, I'll be talking about how you can find remediation. How can you find some way to remediate plastic from the environment? So because of the plastic pollutants or the plastic pollution, one can uh, assume three different ways, three R's: reduce, reuse, and recycling. Reduce, for plastic it's difficult because plastic in every aspects of our life, but we should try to reduce the use of plastic whenever possible. Whenever you can find some alternative to use another utensils except plastic, use that. But again, it is, pardon, any question? And
Any
then,
questions, anyone?
thank
No,
you.
sir. No, sir.
So you can also reuse the plastic. Try to reuse plastic bags, say for example, more than one time, as much as you can. And that's how automatically you can reduce the use of plastic. And then recycling. I know that plastics are recycled in many different ways and different industries are coming up. That is good, but still there is problem. It is not uh, profitable that much because it is very labor intensive, and also the labors that are associated with the recycling industry, very frequently they got some diseases like skin diseases, like asthma. So it's not uh, popular, although as I said, the recycling, plastic recycling is coming up in a big ways. And the other way, you can remove not only plastic but also any other solid waste to burn. So burning is the last type of uh, uh, way you can follow to remediate. But burning is not that we want, because burning of anything releases host of poisonous gases. So you want to get rid of one kind of pollutant which is generating a different type of pollutants. So it is not the solution. Burning is not the solution to get rid of this solid waste. So therefore, you got to find out some other eco-friendly way to remediate solid waste from the environment. And the bioremediation means the remediation through some biological, by the application of some biological agent like microbes, like bacteria, like fungus, etc. And if you properly identify microbes, then microbe mediated degradation can be applied in situ, making the process cost effective and non-laborious, and of course eco-friendly. In most of the cases, microbial degradation is hazard free and yields non-toxic breakdown products. That's how it becomes eco-friendly. So now I will talk about how you can find out microbes for your remediation of target contaminants. So over here, I'll take the one such plastic, polyurethane, but you can apply the idea for many different waste, solid waste or liquid waste. So the idea is very simple. If you want to get or identify some microbes capable of removing your target waste, so the soil or the water sample from the waste site, that would be a good source of microorganisms. So collect the soil sample or some liquid waste from dumping ground and then make serial dilution. Spread it on a plate that contains your waste as the only, your contaminant as the only source of carbon. So if you don't supply any other carbon than your contaminant, and then after several days of incubation, if you see growth on that plate, that means the colony that you are getting, the organism associated with that colony is capable of utilizing that contaminant. So over here, it is the P word. So the agar plate containing the P word as the sole carbon source. So the colonies that will come up on the plate, they will be capable of utilizing P word as the carbon source. So it's up to you, whatever is your contaminant, and then you set up this experiment and find out colonies or the organisms that are coming up on the plates. So over here you can see, so in this plate you can see a clear zone surrounding this holes. So we have isolated some organism or organisms, microbes, and then put those microbes on these holes. And after like 36-48 hours of growth, you can see this clear zone, because these organisms, they are capable of utilizing this plastic material and therefore making this clear zone. That means they are utilizing the plastics, and that's how it becomes transparent. On the other hand, you have a negative control, E. coli. E. coli doesn't have the ability to utilize this plastic material. So whenever you are designing some experiment, you should have negative control, at least negative control. And if you have a positive control, then put it on the plate so that you can understand your experiment is going in the right direction. Then after having the target bacteria or target fungi, so first of all you need to confirm that yes, you have the right organism. So then you can take the dry weight of the contaminant. Over here you can see, so if you have the dry weight, over here it is expressed in breakdown in percent. So the isolated organism can use like 50% of the total solid waste. So it's just by looking into the mass, or if possible, you can look at the morphology, change in morphology of the contaminant. Over here, since we have used a pure film, then we can look at the surface and you can see the alteration of the surface topology. It does indicate isolated organism is capable of degrading the plastic, whatever I am using over here. And the kind of experiments you need to perform, that depends on your target contaminant. So this kind of experiment maybe will not be possible for your target, but I'm just giving you the idea what kind of experiment you can do. And there is next thing. So after the experiments for isolating bacteria or fungi for the degradation of your target contaminant, you may come up 10, 20, 30, 50 different organisms in your hand, and then you can make a consortia, say for example having 10, 15 different microbes together, or you can work with the individual organisms. But most of the cases, it is the consortia that works better than the individual isolate. Over here you can see, I'm just showing an example. When I'm using over here a community, I can see almost 50% degradation. But whenever I have used individual candidates, you can easily see that the community performs better than the individual one. So you got to do experiments to establish or to understand which one would be better. Sometimes you may have one candidate, say for examples, after this experiment, what I have got an individual isolate that has the ability to degrade like 70% of the pure film. Okay, so that is individually works better than the community. So you got to do the experiment which to know whether the community or the consortia will work better or the individual candidates will work better. Okay, then so right now you have the microbes in your hand, either in the community format or in the individual format, that can degrade the target contaminant. However, one thing I should mention, you cannot use that isolated microbes directly in situ, directly to the soil for the degradation or the removal of your target contaminant. The reason behind it, the organism that you have isolated, that is in the lab, right? Now, if you put that organism in the soil, it can destroy the soil ecology. So, you are removing your target contaminant, but at the same time, you are destroying the soil health. And that is actually devastating. So, you must not do that. So, before the application to soil, you must need to check whether if you apply your isolated organism to soil, that retains the microbial health, soil health, retains the microbial diversity. If it fails to maintain the soil health, then you must not apply that isolated organism to the soil for the bioremediation. Although you have the data that it is capable of say 80% removing 80% of your contaminant. Okay, be careful of using microbes in the soil. So then what you can do, alternatively, you can identify the gene or genes that are responsible for degrading that pollutant. Okay, once you know that this particular gene is responsible for the degradation, then you can clone that gene into bacterial expression system and make, say for example, you can use E. coli bacterial expression system and then make that recombinant E. coli capable of degrading your target contaminant. Over here, you can see we have identified the purease gene. We have cloned the purease gene into the bacterial E. coli system. And now you can see over here the recombinant E. coli, whenever it is induced for this enzyme, now it becomes capable of degrading the organism. So the recombinant organism, E. coli, which is not harmful to the system. Now, but again, you never know. After making this recombinant strain, it may be harmful to the soil. Again, you got to test it. But once you have this recombinant strain, then you can purify the enzyme and then apply the purified enzyme for the remediation of your target contaminant. Okay, so you can isolate bacteria or fungi, then test the soil health by some experiment. If it is okay to use to directly to soil, you may have planned to use directly, or even you can go for making some recombinant system, recombinant strain that can express the gene that is responsible uh, for remediating your target contaminant, then purify that protein from the bacterial expression system, purify the enzyme and apply it according to your need. So in that way, you can target to remediate, bioremediate solid waste. Okay. If you have any question, you can ask me at this point. If you so I can proceed
Yes,
now.
sir.
Okay. One more thing, you can also manipulate your isolated strain for better activity. Say for example, over here that strain that I have, I am just manipulating that strain by adding some mineral oil, because uh, this is some hydrophobic surface. The polymers are mostly hydrophobic in nature. So if you add mineral oil, if you increase the surface hydrophobicity of the organism, then you are helping better attachment. So better attachment, better biofilm formation, and better degradation. So you can see over here in the graph, if you add mineral oil in the control experiment, you can achieve. So for this one, the polymer is LDP, the polythene bag. So the organism can degrade like 20% in 30 days. But if you add mineral oil in 15 days, not 15, less than 15 days of time, it can degrade like 20%. And in 30 days of time, it can uh, degrade better. It's like around 24%. So again, you can simply some applying some physical method, you can enhance the degradation. One can also apply the ultraviolet light for plastic degradation. So prior to the exposure to microbial degradation, if you apply some physical method, then make it more susceptible to bacterial or the microbial degradation. That will enhance the degradation process. So that's how, again, you can manipulate the degradation process. Also, look at the biofertilizer. So the application of chemical fertilizers causes, it's not chemical fertilizers, they are not eco-friendly. They damage the microbial soil health, the microbial biodiversity gets reduced, and moreover, the neighboring ponds, they will undergo eutrophication. So the application of chemical fertilizers is not at all good for the soil. So what is the alternative? Alternative is biofertilizer, wherein, say for example, a soil is deficient in nitrogen, a soil deficient in phosphate. So you can isolate organisms. It could be fungus, it could be bacteria again, that are capable of fixing nitrogen, that are solubilizing phosphate, and then apply those microbes to the soil. Again, you got to remember that these biofertilizers should maintain soil health. Otherwise, it could be very devastating. So, for the remediation, for the increasing the soil fertility, you can you can apply microbes, but you must be careful enough so that the application of microbes to the soil, to the environment, doesn't destroy the ecosystem. Okay. So, in conclusion, the spectacular metabolic versatility of bacteria and fungi can be exploited for environmental bioremediation. Microbes can clean up large variety of hazardous substances, chemicals, by transforming them into forms that are harmless to people and the environment. And also one can do some genetic manipulation that can offer a way of engineering microorganisms for better management of pollutants. So that is about solid waste. So right now I'll be talking about, so up to this point, what I have discussed, that is how you can remove or how you can reduce the contaminants from the ecosystem. Now I'll be talking a little bit on waste to wealth. So it is not only you are removing waste, but also you are also generating wealth. East Kolkata wetland, maybe some of you are aware of the East Kolkata wetland and its utility. It is a perfect example wherein waste is converted to wealth. It's a natural setup wherein the city sewage is converted, city sewage is purified, and then city sewage, the purified city sewage is utilized for pisciculture, for irrigation, etc. So the nutrient that is trapped in the sewage, after the purification, they are being released, and with these nutrients, local farmers, they are uh, culturing fish, and that generates huge amount of fish per day without providing any kind of outside nutrient. So let's talk about it. So population of Kolkata is near about 15 million. Daily water supplied by KMC 400 million gallons, and total amount of sewage generated per day is 300 million gallons. Part of this sewage is purified at East Kolkata wetland. These wetlands are located at the eastern fringe of the Kolkata city, and this is the picture that depicts whatever uh, is occurred for the sewage. This is the day zero sewage, pitch black, After day 7, you can see the transformation, and then on day 10, it becomes crystal clear. So that means this pitch black colored sewage is transformed into crystal clear water after 10 days of treatment at the East Kolkata wetland. So these pitch black colored sewage, this is rich in organic waste, right? So you can imagine it has immense amount of nutrients, but they are in unavailable form. So whatever East Kolkata wetland perform, East Kolkata wetland through the activity of the different microbes, it releases that bound trapped nutrients. So that's how it becomes crystal clear, and this water, purified water, is absolutely rich in different macro and micronutrients, and that's how the pisciculture yields per day, it's like 2.48 lakh quintals of fish per day. Imagine it, 2.48 lakhs quintals of fish per day. So that's how, if you think this is waste and now it is transformed to wealth. So this is the natural system, and nature teaches us in this way. You can make waste to wealth. So look at this, the several water quality parameters that has been changed from day 0 to day 12. BOD has been changed a lot. Dissolved oxygen. So at the day 0, no oxygen, but after 9, 10, 10, 12 days, you can see plenty of oxygen present there. Fecal coliform, it's removed almost 99.9%, and that's how it becomes uh, uh, ready to use for pisciculture. It can be used for irrigation purpose. So the trapped nutrients are now becoming usable. So the waste is converted to wealth by natural process. So now what we can learn from this, nature can do this. So can we do this kind of job? So that's how I will tell you the two different things. So renewable energy source, the one of the important renewable energy sources right now, people are thinking in the direction of generating more and more bioethanol. Government has also focused on this. So bioethanol is simple ethanol. Besides the beverage industry, it can right now coming up in a big way for the alternative of renewable energy source, because the fuel uh, uh, like petrols, they are gradually diminishing, and we are facing a time whenever we don't have any fossil fuels. So the alternative source of energy is a big thing right now. So to this direction, bioethanol is very important. So bioethanol can be generated through fermentation, and through fermentation, depending on the type of organism, lactic acid can be produced, ethanol can be produced. So it depends on the type of organism you are using. And what are the resources? Different biomass. Biomass can include like food waste, residues of crops, non-edible crops, algal biomass, etc., etc. So once you have good amount of biomass, If you go to the market, you can see lots of vegetable waste. Yeah, we are one of the biggest producer of potatoes. So, and we may generate huge amount of rotten potatoes. We are we produce very good amount of sugar cane. So this sort of this sort of starchy material can be used to generate bioethanol. So that this is the general scheme of producing bioethanol: biomass, then hydrolysis, then fermentation, and fermentation leads to production of bioethanol. So the two important steps, that is the hydrolysis and fermentation where we can focus. Because say for example, if you have the waste potato, if you have the rotten potatoes, or if you have the vegetable waste, then you got to have monosaccharides to start the fermentation. So they are in polymeric form. It could be cellulose, it could be starch. So you need the help from enzymes that can digest these polymeric starch or polymeric cellulose to monosaccharides, and then monosaccharide again will be converted to ethanol by fermentation, through fermentation. So the monosaccharides like glucose, through glycolysis it can generate pyruvate, but after pyruvate, if it is respiration, it can enter to the TCA cycle and produce carbon dioxide and ATP. Alternatively, if it does undergo fermentation, again depending on the nature of organism, the pyruvate can be converted to acetaldehyde, and acetaldehyde can be converted to ethanol. So you need some, excuse me, you need some enzymatic manipulation. First of all, you have some waste that can be converted to monosaccharides. For that matter, again, you got to take help from microbes. It could be bacteria, it could be fungi equipped with the proper enzyme that can give you the polysaccharides to monosaccharides. Okay. And then once you have monosaccharide, mostly Saccharomyces cerevisiae is responsible for giving you ethanol. But Saccharomyces cerevisiae is also capable of respiration. So if you, it's just an example. If you want to get ethanol, you got to get rid of this pathway. So you can genetically manipulate that particular organism to shut off this pathway, so that glucose pyruvate, and it will be channeled to glucose pyruvate and then ethanol. So you can play around a lot. Say for example, alcohol dehydrogenase is responsible for converting acetaldehyde to ethanol. Excuse me. So if you overexpress alcohol dehydrogenase, then conversion from acetaldehyde to ethanol will be much better. If you can reinforce glucose to enter the cell, so more is glucose, more will be ethanol. So you can express glucose. Glucose cannot enter the cell by diffusion. It is transport mediated. So if you can express more glucose receptor, glucose transporter, then excuse me. So then more glucose will be available for the fermentation. And if you have more ADH, then you will have more ethanol. But even if you can generate more ethanol, Again, you know that ethanol is toxic to any organism. So then again, you have to play around with the resistant ethanol resistance of Saccharomyces cerevisiae. Normally, Saccharomyces cerevisiae can tolerate like 10% ethanol. Sorry for this. So then again, so in one hand, you can play with the production of ethanol, and on the other hand, you can play around with the resistivity to ethanol. And that's why you can get some strain that can withstand, say, for example, as I said, 10% ethanol, maybe you can manipulate it to 30%, 40% ethanol that has some industrial value. So, since we are producing plenty of waste, you can go to the market, you can see it, you can look at your own house, you can see generation of lots of waste, vegetable waste. Then if you plan to make some ethanol, uh, then you can play around with the organism, you can play around with the different metabolic pathways to achieve your target. In that way, you can contribute to the waste to energy production. Another important thing, and that is coming in a big way, that is a microbial fuel cell. It is a potential source of renewable energy. Again, over here, the waste materials are being used directly to generate energy. How? So over here, one can use certain anaerobic organisms. These anaerobic organisms has the ability to eject electrons from inside to outside for their survival. So during metabolism, these anaerobic organisms, they eject electrons to outside extracellular space. And now if you are capable of capturing these electrons to electrodes and then operate through a cycle, through a circuit, then you will be able to generate electricity. So this is a pictorial presentation of microbial fuel cell. It has got two chamber, cathode chamber and anode chamber. Anode chamber is anaerobic, wherein the microbes, anaerobic organisms grow. That means you got to maintain anaerobic condition in this chamber, and you got to add some anaerobic organism. And as a nutrient, you can add waste material, whatever it could be. It could be like uh, wastewater, sewage, it could be the material made from different vegetable waste, etc. But all you need to maintain anaerobic condition in this chamber. And this is aerobic, wherein oxygen is present and half of the tank is filled with water. And these two chambers are separated by a specific membrane which is permeable to protons. That should be very specific. It does not allow the oxygen to enter from cathode to anode. However, protons can be coming from anode to cathode. So during metabolism, these microbes releases electron, and that electron is transferred to the electrode. So electron will go like this, and then proton will go in this direction. So electron, proton and oxygen, they will combine to form water and complete the circuit, and that's how you can generate current over here. So the question is, how do you get this kind of microbe? As I said, these microbes are capable of releasing electron to the outside. So when you are selecting these microbes in the media plate, it could be liquid or solid, add some colorful metal like Fe3 plus, which is brown in color. After getting reduced, it will become green. So if you see, if it is solid, if you see green color surround the colony, then you can understand that that organism is releasing electron. Even if you can use, say for example, dichromate, which is orange in color, right? And it will be reduced to green, chromium 5 state, oxidation state, green. But other thing you can also remember that whenever you are using metal at their highest oxidation state, they could be toxic. So it is not advisable to use chromium 7 because chromium is highly toxic. It has microbial toxicity. So that's why you got to play around with it. It's your choice. So over here, I have used Fe3 plus, brownish color, then Fe2 plus, change to Fe2 plus, it becomes green. So it can be easily distinguished just through naked eye. So that's how you can isolate organisms that can eject electrons for their survival. And if you have the perfect machinery to trap that electrons, released electron, then you can get electricity out of that metabolic activity. So over here, this organism, Paraclostridium species AKS46, it can generate 595 milliamps current per meter square of the electrode. It's not, it's very tiny actually, but this is just the beginning. So no one knows. The two things over here, you can using solid waste or liquid waste. So you are reducing the solid waste or liquid waste, and in against it you are getting some kind of energy, you are generating energy. So waste to wealth, right? So these are the several examples I have given you to elaborate or to discuss the sustainable, what is sustainable development and how can you attain or achieve sustainable development from solid waste or liquid waste, or how could you get rid of the waste material from the environment to get a cleaner, a better environment. And then my suggestion is, although sustainability, it's appears a huge word, but sustainability is not a buzzword. It's revolutionary outlook that has the power to make the change and create a future that everybody deserves. So get involved. So try to practice it. That's it. If you have questions, now you can ask me. Okay. Fine.
Yes, sir. Do we have any questions?
Good morning. I just have a question. Do we have any? We generate a lot of e-waste. So, do
Huh.
we have any process of using microbes to degenerate this e-waste?
Again, e-waste, it contains many metals, right?
Yes, sir.
So then you need to understand the toxicity that is arising from those metals. Say, for example, chromium or lead, right? So chromium causes cancer. They are toxic. Now, you can find out organisms. So then again, one other thing you need to understand, chromium has several oxidation state. Which oxidation state is toxic and which is less toxic? So microbes can reduce the toxicity in two different ways. One is it can reduce or alter the oxidation state. So which one is highly toxic to less toxic? In that way, it can convert it, or it can accumulate the toxic metals within itself, right? That's how it can remove the metals from the environment. But you got to be careful. Again, if you apply those microbes in situ, again the metals will remain in the environment. So then you need to arrange something for those microbes accumulating the metals to make it away from the environment. Okay, understand?
Yes, sir. Thank you, sir.
Any other questions? Okay, so to summarize, effective waste management requires an integrated approach, combining technological advancements with ecological principles. Today, Dr. Alok Kumar Shield's work highlights the importance of microbial activity and the need to combine this with other waste management techniques. It is by doing this that we can reach a sustainable future. So, thank you once again, sir, for your insightful talk. We are really honored to have you with us today. Thank you, sir.
Thank you, Payal. So I am leaving, right?
Yes, sir.
Okay.
So, the next session is from 12 p.m. It will be given by Dr. Obrojyoti Ghosh, Associate Professor, Department of Biological Science, Bose Institute. So please join with the link provided in the group. Thank you.